Assalamualaikum. Thank you for watching Dr. Syed Rizwan Abbas YouTube channel. On this channel, you will learn about chemoinformatics, how to drug design by using structure-based drug designing, ligand-based drug designing. Only the studies regarding to drug designing you can learn from this channel. And today in lecture, we will discuss about the basic steps of chemoinformatics. Because if you want to come in chemoinformatics and you having a background of biological sciences, life sciences, computer sciences, medical sciences, you must know the basics of chemoinformatics. Then you will be able to work on drug design. Other than it is very difficult because many terminologies are using in chemoinformatics without basic information or strong background. Without strong background it is impossible. So in today's lecture we will discuss a little bit about chemoinformatics just for information and for your kind understanding. Difference between molecules or compounds, leads compounds, ligands, pharmacophore. These are four steps. Today we will learn about what these are actually. The first one is the compounds. Compound everywhere we are using, we having interactions with compounds in daily lives. A compound is a material formed by chemically bonding two or more chemical elements. Because you are the science students, you know about the elements, about these basics. The type of bond keeping elements in a compound together may vary. Covalent bonds and ionic bonds, they having different bonds, maybe covalent or ionic, are two common types. The elements are always present in fixed ratio in any compound. For example, water and berberine. Such type of molecules, type of compounds in our lives are present in millions, in billions, everywhere in our lives. We have seen, we have interactions, and we are eating, we are wearing, we are looking, everywhere are the compounds. So from the compounds, we screened like drugs, pharmacophores, from all these compounds, from the compounds, like the basic of chemoinformatics comes from compounds or molecules. So leads compounds comes from compounds, molecules, the basics, and from that, after the screening, we screen some lead compounds. In medicine, a chemical compound that shows promise as a treatment for a disease and may lead to the development of a new drug. Thousands of compounds are tested to find a lead or leading compounds that may act on specific genes or protein involved in a disease. So for that process, from compounds, there are millions and billions. So from that, we screened some specific compounds by using some parameters, which parameters we will discuss in detail in coming lectures. By using the parameters, we screen some basic screenings are required to screen out from thousands, from millions compounds, only in hundreds. Like how many compounds are eligible to move ahead as a drug. So we can select, we can screen some compounds. That's why these compounds are known as leads compound, because they are the leaders now. They come pop out. That's why these compounds known as a leads compound. From leads compounds, is further screening is required. These are known as ligands. What are the ligand? Molecules that bind to the receptor. To a receptor are called ligands. The binding can be specific and reversible. A ligand may activate or inactivate a receptor. Activation may increase or decrease a particular cell function. Each ligand may interact with multiple receptor subtypes. So, after the screening of lead compounds, further screening is required for ligands. So what will happen when we screen some lead compounds after the screening of its basic parameters, they are eligible for a drug or not. So they become the lead compound. And further, the next step is how to screen the ligands from lead compounds. For that purpose, we used protein docking and some other methodologies. So we check the compounds, how much they having the capability of the efficacy to activate or deactivate of the compound of the receptor, like the proteins or the enzymes which we have the target. Like for example, if we want to inhibit any proteins of a specific disease, so for the docking for the inhibition of the specific proteins, we required molecules, and we required some compounds. These compounds are become like the ligands, and from ligands we screen some molecules or the compounds from ligands for the further screenings and check how many compounds or the ligands having capability to dock our target protein. This is called ligands, which become the ligands now when we get some best efficacy score between the ligand and the receptor. So we finalize these ligands are the best. And at the final stage, when we screen the ligands, these ligands become a drug. And further, we do more studies, like a precise work is required. Furthermore, for example, any substance other than food that is used to prevent, diagnose, treat, or relieve symptoms of a disease or abnormal conditions. Drug can also affect how the brain and the rest of the body work and cause changes in mood, awareness, thoughts, feelings, or behaviors. So it means the drug, for example, you get headache and you go on any medical store or a doctor and you get some painkillers, and after getting this you relax, some feel like better. Why you feel better? Because drug effects. So same like this, the ligands which we finalize it, they having the capability to dock a target proteins which we wanna, but we haven't the research. After that, if we get two or three molecules, so on two or three molecules, we do further studies like wet lab trials, biochemical assays, animal trials, and it's moved to FDA for approval. So the finally it's become the drug. This is a simple procedure for the drug development by using chemoinformatics. So the summary of today's lecture is very interesting from that you can easily understand. Compounds are molecules. So further, the first one are the compounds are molecules. Millions are present. We do some models like QSCR model and many other models, admit properties and many other screenings, molecular weight, TPSA, log P, and some many other parameters. Precise parameters are used to screen is specific ligands from millions of the compounds. So they become the ligand compound in second steps, like from millions, from lakhs, like in thousands. After the using some parameters, it's we screen near about 100 in compounds. These 100 compounds become ligand compounds. And for the further screening, docking are used. Docking are used on hundreds of the lead compounds to screen for the screening of ligands. These are the ligands after the screening by using docking of target proteins of the receptors which we have. And all the ligands when we finalize it like the best 20 plus final trials it's moved. If final trials means like animal trials and some other wet lab trials, toxicity test, its adverse effect, side effects, we check and dosage, each and everything. Of course, we use mostly some parameters by using in silico studies we can screen, and furtherly and finally, of course, it's required the wet lab and human trials, etc. And it's moved to for the final FDA approval, and it's become the drug, and finally it's drug release. So all these methods are used by like using in silico studies. We can use it within a time. And Today lectures we learn about the basics of chemoinformatics, like one part of our chemoinformatics about the molecules, about the compounds, how we select where from we get the molecules which are used finally as a pharmacophore as a drug. So this was today lecture. Thank you very much. If you have any query, you can send me email on this email number. I'll put down in comments. I will try the best answer of your question. Thank you. Thank you very much.